Hi, welcome to all. In this video class, we are going to study the topic reporter gene assay. Reporter gene assay. We already studied the process of DNA transfection. We know that DNA transfection is the process of transferring DNA fragment or gene into the cell deliberately in order to create genetically modified organism or genetically modified product. When we come to the topic reporter gene assay, it is a gene which enabled the measurement or detection of a specific foreign gene within the cell. If you want to check, suppose if you want to check the presence of a foreign DNA inside the cell, if you imagine, if you insert a foreign gene into the cell, after that we need to check whether that foreign DNA, that inserted foreign DNA is present inside the cell or not. For that purpose, we can use reporter gene. That is why this topic is called reporter gene assay. So the main features of this reporter gene is that its gene product or its translation product can easily detect by using different kinds of technique. In this class, here we are going to study what are the examples for, what are the different types of reporter gene which are used in genetic engineering to analyze the presence of a foreign gene inside the cell. Normally, these reporter genes are ligate or tagged on a foreign gene which is going to be inserted into the cell. Therefore, we can easily able to analyze or we can easily able to understand the presence of a foreign DNA inside the cell. So the main use of this reporter gene is to detect the presence of a foreign gene inside the cell. The foreign gene is whether present in the cell or not. So for that purpose, we are using reporter gene. So that technique is called reporter gene assay. Let us take, and we have in biotechnology we can find out lot of different we can find out different kinds of reporter gene. So here we are going to study few examples for reporter gene assay. First of all, let us move into the first reporter gene, that is GFP. GFP means it is a reporter gene, that is green fluorescent reporter gene. GFP means green fluorescent reporter gene. That is green, G for green, F for fluorescent, P for protein, green fluorescent protein reporter gene. First example for reporter gene is a green fluorescent protein reporter gene. This GFP reporter gene produces a protein. That protein is called green fluorescent protein. And that green fluorescent protein consisting of approximately 238 amino acids. It is made up of 238 amino acids with a molecular weight of 26.9 kilodalton. That is the main feature of this green fluorescent protein produced by GFP gene. And the salient feature or the main feature of this GFP protein is that they can able to perceive light with a wavelength of starting from violet to UV. So it mainly absorbs UV light, not accurately UV, violet, a light wavelength ranges from violet to UV. And after absorbing that wavelength, it shows fluorescent property in the form of a green light. That is why it is called green fluorescent protein. So after absorbing UV light, this GFP protein shows fluorescent property, that is it gives after absorbing UV light, it gives green color fluorescent property, that is it shows green fluorescence. So by using this reporter gene, if we tag this GFP reporter gene along with a foreign DNA, we can detect, we can easily understand the presence of foreign DNA by this property, that is showing green fluorescent property. So it gives green fluorescence. When we tag this gene along with a foreign DNA, then after entering that foreign gene in the cell, it gives green fluorescence. So we can easily able to detect with the help of our naked eye or using small microscopic instruments. That is the main feature of this green fluorescent protein. And second example for reporter gene assay is called Luciferase gene, LUC gene, that is Luciferase gene. This Luciferase gene is found in different living organisms. For example, it is found in few bacteria, few insect like firefly. In Malayalam we are saying minna minni. And also found in some other marine organism, that is jellyfish. So it is commonly found in more than one organism. Bacteria, firefly, jellyfish. This Luciferase gene produces a proteinaceous enzyme. That proteinaceous enzyme is called a Luciferase. That is the main features of this LUC reporter gene. It produces an enzyme called a Luciferase. It is found in bacteria, firefly, and jellyfish. And let us move into the bacterial Luciferase. This bacterial Luciferase catalyzes an oxidation reduction reaction simultaneously. That is why that reaction is called, that is why it is called oxidoreductase. It is called oxidoreductase. Bacterial Luciferase is an example for oxidoreductase because it catalyzes both oxidation reaction and reduction reaction simultaneously. That is why it is called oxidoreductase. Here you can see that oxidoreductase reaction. Here you know this is FMN, that is flavin mononucleotide in reduced form, that is FMN along with the H2, that is reduced form of flavin mononucleotide. Okay. It reacts with molecular oxygen and aldehyde, CHO aldehyde, in presence of bacterial Luciferase. So this bacterial Luciferase convert this reduced form of flavin mononucleotide into an oxidized form of flavin mononucleotide. So it is an example for removal of hydrogen or addition of oxygen. It is an example for oxidation. Like that, it also converts molecular oxygen into water molecule. Is also an example for reduction. So here both oxidation and reduction reaction takes place, but the significance of this reaction here is that during this reaction it gives light. It gives light which can easily able to detect with the help of human eye. So that is the main features of this bacterial Luciferase. So if we tag this bacterial Luciferase along with the gene which is going to be inserted into the cell, after the insertion of that gene, we can easily able to detect the presence of that gene by detecting the light emission due to the presence of this tagged bacterial Luciferase along with that gene. So it is also an example for reporter gene assay. Like that, this Luciferase is also present in marine organism like jellyfish. That is called jellyfish Luciferase, LUC gene, reporter gene. The main features of the main function of that Luciferase is that it converts the reaction. Here that reaction is called Luciferase acting reaction is called luciferin. So here jellyfish Luciferase convert the compound luciferin into an unstable intermediate through oxidation reaction. That unstable intermediate then converted into oxyluciferin. Luciferin undergoes oxidation and form unstable intermediate, which then converted into oxygenated luciferin, oxyluciferin, by releasing light, which can be able to visualize by using naked eye. So this can also be used as a reporter gene to check the presence of a foreign gene inserted in the cell. So this is an example for reporter gene. When we move into a third type of reporter gene, that is called cat gene. This cat gene produces a protein. That protein is called chloramphenicol acetyltransferase. Cat gene produces an enzyme, a protein that is enzyme that is called cat enzyme, chloramphenicol acetyltransferase. The presence of that chloramphenicol acetyltransferase gives the organism to resist against the antibiotic chloramphenicol. So if we ligate this reporter gene, CAT gene, along with foreign DNA which is to be inserted into the cell, after the insertion, we can check its presence by culturing that genetically modified organism in a chloramphenicol antibiotic containing medium. If it resists, then we can say that foreign gene is present inside the cell. So here CAT gene is used for detecting the presence of a foreign gene inside the cell. When we move into the fourth type, we know that fourth type of reporter gene is called LacZ gene. These LacZ genes are usually found within the operons of lactobacillus, LacZ. LacZ is a gene which produces an enzyme that is called beta-galactosidase. That the main function of that beta-galactosidase is to cleave galactose. So that beta-galactosidase can be used as a best reporter gene. The presence of beta-galactosidase, if we ligate this beta-galactosidase along with the gene which is going to be inserted into the cell, then this beta-galactosidase gives the bacterial colony to a somewhat blue color. So if the recombinant bacteria carry this reporter gene along with the foreign gene, it shows blue color. If the reporter gene is absent, then the bacteria gives only colorless, that is white colony. So the presence of blue color gives blue color. The presence of blue color says that bacteria contain foreign DNA or foreign gene with this reporter gene. And when we move into the fifth type of reporter gene is called GUS reporter gene. This GUS reporter gene produces an enzyme. That enzyme is called beta-glucuronidase enzyme. So beta-glucuronidase enzyme producing gene is called GUS reporter gene. The main function of this beta-glucuronidase is that it converts glucuronides. That is glucuronide. For example, I'll write one glucuronides here. 5-bromo-4-chloro-indolyle glucuronide. It's an example for glucuronide. So this beta-glucuronidase convert glucuronide, that is for 5-bromo-4-chloro-indolyle glucuronide, into a product which gives blue color. So it also considered as a reporter gene because its gene product shows blue color which can be easily visualized. So here we discuss 5 types of reporter gene assay, that is GFP, green fluorescent protein gene, Luciferase gene in bacteria, firefly and the jellyfish, chloramphenicol acetyltransferase cat gene, beta-galactosidase gene, GUS beta-glucuronidase gene, like that. We have different kinds of reporter gene in plants and animals. Here we discuss only 5 types. This is the broad concept regarding with the reporter gene assay. In simple terms, it is a gene to check the presence of an inserted DNA or gene inside the cell or not. For that, we ligate or tag the gene of interest to be inserted into the cell along with this reporter gene, so that we can able to trace or we can able to detect its presence in the host cell. That is the concept. Thank you.